Good afternoon to everyone. Thank you for the introduction. So today I'm going to be talking about the coupling of dark fermentation and microbial electrolysis cells for hydrogen production and the microbiome involved. So the index that I will follow during the presentation, I will start with a brief introduction and then describe the material, methods, and the results that we obtained, and then to state the conclusions of the work. So, well, my colleagues, I'm the last one from the LBE team, so they already explained the fermentation process. So I'm going to skip this part, and I'm going to go to the part that justifies the coupling of the dark fermentation process with the MEC. So as Eric mentioned this morning, the yields in the dark fermentation are low. So for instance, considering the acetate pathway, we can only obtain up to 4 mole of hydrogen per mole of glucose. And this is only theoretical because then the experimental yields are quite lower. So most of the organic matter remains in form of the volatile fatty acids, which can serve as a carbon source for the subsequent MEC process. So like this, we could theoretically increase the theoretical yield up to 12 mole of hydrogen per mole of glucose. And also in the MEC step, we could recover hydrogen, which is much purer than in the dark fermentation. However, the process, this coupling, relies especially on the volatile fatty acid composition that we impose in the MEC, and also on the microbiome that it is developed in the cell. So the specific objectives of this investigation were to identify the dark fermentation conditions that shape the volatile fatty acids profile, to assess the hydrogen production in the MECs, and lately to analyze the microbial community developed in the electrode. So as inoculum and substrate for this experiment, we used an aerobic sludge, which we freeze-dried and pre-treated at 90 degrees for 15 minutes. And we used the same model food waste that Lucia showed in the last presentation. So we grinded and sieved this food waste, and we pre-treated it for 1 hour at 70 degrees. So then we carried out the dark fermentation in 2 different reactors, in flask bottles and in reactor vessels of 0.5 liters and 2 liters, respectively. And we analyzed the volatile fatty acids composition that we obtained and the biogas in our headspace. And finally, when the dark fermentation ended, we centrifuged this effluent, and we used it as a media for our MEC. So the MEC was a dual chamber of 0.8 liters. We carried out this experiment at pH 7. The anode was made of carbon felt and the cathode of platinum. And then as a separator, we chose a cationic exchange membrane. So how we did this experiment is that we enriched the anode using a synthetic media and acetate as carbon source, and then sequentially and in the same reactor, we added the different effluents that I will show you afterwards. And then between processes, we took samples for Illumina sequencing. So going into the results for the first objective, which it was intended to identify the operating parameters that shaped the volatile fatty acids profile, we observed in the pH screening from values from 5 to 8 that we had 2 different behaviors. So on one hand, we had the acidic pH values in which we obtained a high butyrate concentration and high hydrogen. And then we had the other behavior, which was at pH neutral, at neutral pH and pH 8, in which we obtained a higher conversion between 50 and 65%, and acetate was promoted in these effluents. So conversion, we define the conversion as the amount of metabolites that we obtained out of the total organic matter that we fed into the system. So here we observed these 2 different behaviors, so we decided to carry on with our experiments by selecting the neutral pH, and then between the acidic range, we decided to go for a pH in between 5 and 6, because there are a lot of studies dealing with dark fermentation at pH 5.5, so we thought this would be convenient for comparison purposes. So with these two pH values, we went for the temperature screening from 25 to 50 degrees. And what we observed was that the best temperature that promoted hydrogen production with a similar conversion that we obtained before was, again, the 37 degrees. So we decided to carry it on with this temperature and these two pH values. So afterwards, as I mentioned before, we enriched the bioanode in the MEC. And we decided to start with the pH, the effluents from pH 7. We fed it by duplicate, and sequentially, and then the ones from pH 5.5. So I'm going to show you the results of the first one and the last one, but these results are similar also for the duplicates, so the behavior, we can extend it to the duplicates. So first of all, what we observed was volatile fatty acids removal higher in the effluents coming from dark fermentation at pH 7 than the ones coming from the fermentation at pH 5.5. We observed as well that acetate was totally consumed, and it was preferentially consumed compared to the other volatile fatty acids, such as here the red one, propionic, or here the butyrate. And as I just mentioned, butyrate was partially consumed, but it remained unconsumed at the end of the experiment. So this, of course, impacted the overall yield of the process. And we observed global hydrogen recoveries way higher in the effluents coming from the dark fermentation at pH 7 than the ones coming from the dark fermentation at pH 5.5, which were below 10%. But we wanted to further know why this happened, as we observed volatile fatty acids consumption in both effluents, but this was not extensible to the total hydrogen yield observed. So for that, we went to analyze the microbiome that we had in the electrode. First thing that we observed was that the Shannon index was a bit higher in the electrode than in the bulk phase, and it was quite similar for all of the stages. This, we attributed it to the fact that we use an electrode made of carbon felt, and this is a material that it is highly porous, so part of the biomass might have been entrapped in there, contributing to this index. Then with regard to the dark fermentation effluents, both of them, from pH 7 and pH 5.5, they had a high relative abundance of Clostridium species. And then moving into the relative abundances that we observed, we have the bulk and the electrode. So I'm going to focus on the electrode especially. What we observed was that the low acetate ratio negatively impacted geobacter presence. So as you can see in these first two stages with the effluent from pH 7, the geobacter relative abundance was between 30 and 40%, but then when we changed the effluent, it decreased to values below 20%. And also what we observed was that there was a development of species such as Clostridium, Petrimonas, or Azospira, which were not correlated with the hydrogen production. So I think before there was a question about the existence of fermenting microbes with the electroactive bacteria. So in our case, we had this coexistence. And by the way, this might explain the lower hydrogen yield obtained in this when we use this effluent, because this species might have used the electrons, instead these electrons being transformed to hydrogen. So as conclusions for this work, we say that the process pH in the dark fermentation shaped the volatile fatty acids profile because we observed great acetate abundance with the pH 7 and butyrate with pH 5.5. Then when we applied this influent in the MEC, we observed a consumption preference. First was acetate, then was propionate, and finally it was butyrate, which remained unconsumed probably because the concentration was too high. And then the poorer hydrogen yield obtained at pH 5.5 was attributed to the fact that geobacter relative abundance was lower than in the first effluents, and the colonization of the anode by other species that didn't contribute to the process. So future studies should address in this sense the capacity of the bioanode to electroactively consume butyrate. So thank you for your attention.